Biopharma 4.0 has made significant strides since the release of the FDA and EMA guidelines on process analytical technologies nearly 2 decades ago. The mission has always been to push towards predictive control and ensure quality by design, and a significant player in this journey has been multivariate optical sensing technologies, showing immense potential for inline continuous monitoring. The allure of these technologies lies in their ability to provide hands-off, non-destructive, and easy integration into multi-parameter analysis. However, they also bring their own set of challenges, which we'll address today. I'm Miles Skoda, and this is the Life Science Connect webinar series, and our topic for today's session is inline monitoring of bioprocess parameters for the 21st century. Now, today we are incredibly privileged to have two industry experts from 908 Devices who will shed a light on the current state of several optical technology approaches and introduce us to an exciting new platform. We have with us today Dr. Christopher Brown, the Chief Product Officer and co-founder, and Dr. Graziella Piras, the Senior Director of Strategic Marketing. Now, we'll be holding a Q&A session following this presentation, so feel free to submit questions as we go along. We'll get to as many as possible during the Q&A, and any that we can't get to, we'll follow up on after the event. Just a bit of housekeeping. If you run into any issues today, please try refreshing your browser first. If your problem persists, submit a question via the Q&A, and I'll help get you back on track. And then finally, this event will be available on demand, usually within a day or two, and you'll get an email with a link to it, or you can just use the same link that you used today. So with all that said, I'm going to turn it over to our experts to get started.
Thank you so much, Miles. So hi, Chris. How are you today?
Yeah, good. How are you?
Excellent. So Biopharma 4.0, process analytical technologies, and guidelines from the FDA said 20 years ago. Can you tell us a little bit more about all that?
Yeah, absolutely. So in the grand scheme of biopharma and pharma 4.0, analytics is really a key part of that, and the use of advanced technologies to guide us toward reaching the biopharma 4.0 objective. In the context of analytics, people always talk about sort of 4 steps through that process and the tools to support those. So there's a descriptive analytics, that is what's happened. There's diagnostic analytics. Why did it happen? Do we understand what's going on? Those two connect to form some basis for predictive analytics, understanding enough about what's happening to know what will happen. And then ultimately, end game is prescriptive. That is, can we actually control our destiny with the input of a lot of analytics inputs, knowledge inputs, and otherwise? And that's really where PAT is a key element of that story. So PAT stands for Process Analytical Technologies. And that term has been around for quite some time, as was noted. It's certainly been available in industry guidance for pharma. But PAT is a package of technologies that encompasses instrument sensing, other data in feeds, and those get processed in such a way that data becomes information. And that's an important differentiation. Data by itself is not information. And that information then becomes a synthesizer for knowledge. So it's beyond the diagnosis of what's going on, but what do we know about this process, and what does it tell us about what to do? And then, of course, the ultimate end game is to use that knowledge to drive action. And that action is a function of process analytical technologies and the analytics they provide, but it's also a function of the greater sort of process of knowledge. PAT specifically has been defined as a system for designing, analyzing, controlling manufacturing through timely measurements, and of course, converting those measurements into, as I noted, information, ultimately knowledge. And it's a cyclical process throughout the development of a pharmaceutical product, and it's a cyclical process in the production of the product in terms of being able to continue to maintain the ability of the process to produce product at the expected yields as well as the expected quality profile. So the interactive cycle of analyze, design, control, and analyze is an important recurring cycle within PAT and analytics in general. There are a number of sensors that are in play in the world of process analytics, certainly in biopharma. So some are widespread, some are limited. In the widespread category of inline sensors and analyzers, a number of attributes are widely available. So pH, dissolved oxygen, temperature, flow, partial pressure of CO2, certainly glucose, lactate, optimal density, and biomass. The technologies there tend to be enzymatic, electrochemical, single channel optical, or capacitance or impedance for those attributes that I noted. There are also limited online analyzers today. Many of them tend to be enabled by automated sampling systems. So they're more sort of traditional offline technologies, but with a sampling system providing sampling. Examples would be glucose, lactate, glutamine, cations, etc., cell counts and viability. And the technologies are quite varied for what can be serviced by automated sampling systems. But those are quite limited. They're quite complicated given the complexity of online sampling systems. And so a little more typically, a lot of those attributes are measured at or offline, so disconnected from the actual process itself. And these are widespread, but they might be a system that's sitting in a laboratory where bioprocess is running, and samples can be brought to that analyzer. So there's a lot of attributes there that are accessible: amino acids, osmolality, cell counts, density, viability, CQAs in general. And the technologies are a little more high-end or a little more broadly capable there, again, because they can service a lot of different bioprocesses. Mass spec, for example, osmometers, labeled imaging, chromatography, electrophoresis, spectroscopy, and cytometry.
Great, thank you, Chris. That was very informative. So at 908 Devices, can you tell us a little bit how you have supported and enabled some of these process analytical technologies and analyzer?
Yes. So we've been very active in this domain and invested a lot at 908 Devices in trying to help people in the Bioprocessing 4.0 initiative. So we have several products that are today in the marketplace. So one is the zip chip down on the lower right-hand side. So that's a capillary electrophoresis microfluidic system that connects to a large mass spec platform, and it supports a very rapid analysis of CQAs and KPIs. So it could certainly do tighter glycosylation in detailed glycobiology, charge variants, full versus empty capsids, etc. It's very high resolution separation and a lot faster than HPLC. The Rebel is an at-line system that's really built for the cell biologist and PD scientist. It's focused more on critical process parameters, in particular amino acids, some vitamins, dipeptides, for example. And then in the ecosystem of more online connectivity, we recently released the Maven at the start of this year. And the Maven is an inline system to do real-time glucose and lactate, used across a wide variety of media types of bioprocesses, and control at a very low level. As a family, then, we have a nice portfolio that we're very proud of. So the Zip Chip, the Maven, and the Rebel. And we're really delighted today to introduce a new member of that family with some really exciting capabilities. So this is the Maverick. It's a Raman monitoring platform for multiple critical parameters, and really importantly for us, it's ready to go in minutes. Very compact platform and designed to do turnkey analysis for glucose, lactate, biomass, and more, but with the simplicity of a pH meter. So Raman systems that have been available and generating some excitement in the marketplace due to the technology capability have really been quite challenging to actually get up and running and configured. Really, Maverick is a lot more of a turnkey system that can be used very simply with the complexity of a pH meter. So no connected big computer system, and a small on-screen touchscreen that reports real-time glucose, lactate, and biomass, and you can access system settings and handle all of the needs required for the Maverick just on the small screen that's attached to the device. Beyond that screen, not everybody hangs out really close to their bioreactor all the time, and so Maverick also has the ability to be remotely accessed from anywhere you've got a web browser. So whether it be on your phone or back in your office on your laptop. So quick summary of Maverick at a glance. So as I noted, turnkey solution for glucose, lactate, and biomass. We've really been focused on mammalian capability with the initial launch of Maverick. So CHO, HEC-293 processes, a lot of validation work there. For glucose, lactate, and biomass, there's no modeling required. So no significant startup period or learning new software or short courses for various chemometric modeling approaches. It has no ambient lighting restrictions, so it can run in daylight, in room lights, or at night perfectly happily. All the compute is completely embedded, so it really is an embedded application, all self-contained and self-controlling. It does have both analog and digital feed control outputs. So on the back of the device, you can use current or voltage to directly control a feed pump, for example, or signal another device. Provides data and outputs via CSV or OPC UA, and it really is built to be 21 CFR Part 11 compliance ready. So you have full support for usernames, password restrictions, account logging, and audit trails supported on the device. The other exciting attribute for Maverick is that you can multiplex up to 6 bioreactors from a single hub. So the top tilted unit we call the hub, and that hub supports talking to up to 6 measurement modules. It also minimizes cords, so there are plenty of cords running around bioprocess labs, power plugs, etc. But Maverick can run just with power over Ethernet, so that's a big advantage for simplicity, and it can talk to those up to 6 different bioreactors concurrently. As I noted, you can get remote access from any web browser. So type in the IP address, and you can jump straight into seeing what's going on with your bioprocesses. And we do make the data available offline, so the raw spectral data is available for any additional modeling that you or your teams might be interested in doing. So certainly feeding that analytics engine that is such a big part of Biopharma 4.0. We realize some folks might want to do more with Maverick than with lactate biomass, and so the data is available offline for you to extend the capability of that platform. It is a Raman spectrometer, so in terms of the details of that spectrometer, it's a 785 system. Output power at the probe is about 450 milliwatts, so it's a class 3B system. Spectral range is very standard, 200 to 3200 inverse centimeters at approximately 6 wave number resolution. It's a cooled CCD system, 1024 pixels, and as I noted, class 3B. It has a very myopic optical lens, though, so there's only a 4-inch optical hazard around the end of the probe, and it's class 1 outside of 4 inches, so no need for over-the-top laser safety measures or glasses or anything like that. As noted, the hub can control and power up to 6 measurement modules. So this is a back view of the Maverick hub. You'll see a number of Ethernet ports there, 7 in particular. So 1 is for connectivity to the customer network, if desired, and the other 6 are for directly communicating with measurement modules. So that's not only data communication, but also power, as noted. CSV, or Excel-style results, can be exported at any time during a process run. The raw data is batched and exported at the conclusion of a run. As noted, OPC UA or analog outputs are present, and the analog outputs are on the back of each measurement module. So each measurement module can control an individual feed controller or signaling system. The full resolution spectral data is in contemporary HDF5 format. That's the sort of NASA NSF standard for high density double precision data. The software was built for GMP readiness, as well as 21 CFR Part 11, as noted. And as a turnkey system with a completely encapsulated capability, it's considered GAMP5 Category 3. So relatively straightforward in terms of product validation.
Thank you, Chris. This was really informative. Thank you. And it does seem like the Maverick is a great tool to implement inline monitoring. You mentioned that basically there is no modeling required on the user side. I understand that Raman, usually using Raman in inline monitoring requires a lot of modeling, chemometric modeling, and it's costly and time consuming. So can you explain a little bit, how you approach this technology for Maverick? Would love to hear a little bit more about it. What's in the box?
What's in the box? Yes. Yeah, so very, very powerful analytical technology in Raman, but it can be a significant hurdle to getting everything up and running. So lots of publications over the last 20 years applying Raman in bioprocess characterization, monitoring, but getting multivariate models to cooperate in a very robust way is quite a lift. Certainly doable, but quite a lift. This was shared sort of by one of our customers, that there is certainly the initial equipment purchase price, and a lot of these are quite expensive. But beyond that, there's also very significant costs in the rest of the process. So there's the software required to do the modeling. It's not particularly cheap. If you're not so lucky as to have in-house expertise already, then there's typically quite a bit of training required to use that software proficiently. But then the bigger kicker comes with actually developing what are usually referred to as appropriate calibration models. And because of bioprocess complexities, it's significant. Most people talk about 12 to 36 months of getting sufficient bioprocess data to actually build robust calibrations. There's a lot of SOP development along the way during that phase. And when you think you've sort of arrived at a robust model for the attributes of interest, then there's still method validation required. Requires data collection, maybe some final optimization and model maintenance that's ongoing. So that whole pathway is certainly supportable by some large groups that have a lot of that capability in-house already. But for a lot of other customers, it's quite a lift to get there. And so when we were developing Maverick, we were very focused on trying to shorten that time cycle. And so one way to look at it is this: with a traditional Raman, you're buying a spectrometer, so you're buying a data collector, and the remainder of the solution is really on the burden of the customer. The startup price is often pretty high, so some of these platforms are quite expensive. So in terms of time and expense, you're already starting with a fairly high sticker price. But then that phase of sort of developing models, the calibration for the system, observing data, collecting more data, there's a long period over which you're collecting data but not really realizing a lot of value from the implementation. And so our goal with Maverick was to certainly reduce the initial startup costs. So we are aiming for a more manageable entry price for the platform, but then also providing that immediate value for glucose lactate biomass, and allowing people to realize value right off the bat while they're still maybe collecting some additional data, and perhaps with some extended modeling offline, they can get further upside from the platform in the future. So trying to really change the value curve with Maverick, with providing a lot of turnkey capability. So how do we do that? And why is it so hard? Why does it take so long a lot of the time? So bioprocesses in particular are a tricky application for developing multivariate models. And the reason that it's so tricky is that bioprocesses are extremely intercorrelated between a lot of confounding factors. So, right, glucose is consumed, waste products increase, cells grow, and the fact that this is really a sort of a metabolic reactor, it means that when things go up, other things go up. When things go down, other things go down. And the tools that are generally used to develop these multivariate models have a very difficult time deconvolving those confounding factors. So, like my images on the right-hand side, it's quite a tangled mess of confounding factors, and it takes a lot of data to ensure that you've sort of broken or deconvolved a lot of those confounding factors. So what's typically done? Well, there's a lot of acronyms in the space. PCR, PLS, multiple components, etc. But hopefully, things start with problem understanding. So, what is somebody trying to achieve? Selection of an analytical platform and some proof of concept experiments. Then you write a check for a piece of equipment, a spectrometer. And then you start collecting data. So it's most commonly observational data from real bioprocess runs. Reference values are collected from an independent analyzer. So maybe you're trying to do glucose lactate, something else. So a reference analyzer is running, collecting that data. And then you try to put the two together. So no matter what software you're using, you're looking at the spectral data, you're looking at the reference data, doing an exploratory data analysis, then building some model which links the spectral data to the reference values. And there are a lot of choices along the way in terms of pre-processing and spectral ranges that are part of that process. And you can look at the performance of that relationship or that model in terms of cross-validation. So how does it do predicting, the process data that we have, and does it seem to be meeting the needs of the customer? And at some stage, you really have to predict. So nothing's of real value if it can't get to that prediction phase that we were talking about in the analytics journey. So what does it do with new processes? And are the results that we observe with new processes really robust, stable, reliable? Is it a useful analytical method overall for us to use? The Maverick approach is quite different, and there's some advantages that we have here that we directly exploit. So we use what's called a de novo model, and de novo meaning more from first principles. So there are some references down at the bottom that have some intimation to this, and we'll be releasing some tech notes as well that detail this more carefully. But at the factory, so we build a Maverick. There's a general element of the de novo model, which is what we call sort of the master formulary. So this is a master list of the whole world of possible media components across all sorts of mammalian, not a specific media, but the actual components of the media. Along with the list is a probability density. So it's really a large matrix that has prior probabilities about the typical ranges over which those media components would be found. That prior probability isn't a really hard bounding phenomenon, so it doesn't matter if one media type has some higher concentrations than another, but it does guide the math that follows to balance out what is expected in sort of the broader world of these cell culture media. So that's a general formulary. On the single device level, as we build Mavericks, there are a couple of other really important pieces. So for a given Maverick, an instrument measurement error model is built. And what does that mean? Well, it's a detailed physics-based model of the spectrometer, the optical path, and the behavior of electronics. And that allows each device to know exactly what the integrity of its data is, what the characteristics of its optical collection are. And the final piece is an optical filter for that spectrometer. So based on the measurement error model and the attributes that we're chasing, based on the master formulary, an optimal filters is defined, and that's different on every instrument. And that's really carved around the spectrometer and the electronics. So we can do that for every Maverick we ship. And that allows it to actually build a de novo model. So it's an embedded capability that is built to predict whatever is turned on at the moment. So glucose, lactate, biomass out of the gate. When it gets to your lab, then the de novo model is essentially ready to run, with the exception of an optical fine adjust with a system suitability and a PQ. So there are two quick system suitability standards that are run that allows it to confirm that it's doing what it should be doing from a PQ perspective. Once that's done, then you can autoclave the probe and hit go. So nothing else to do to get a Maverick up and running in your mammalian process. A picture of that in your lab process, so it's really just 1, 2, 3. So Maverick arrives ready to go. The two PQ standards come in a kit, and Maverick's probes immersed in each of those in succession. That allows it to compute its complete its system suitability. And from there, you can take the probe out of the off the system, unautoclave the probe in your reactor, or the probe can be autoclaved by itself as well. Totally up to your SOPs on that one. And then immerse, of course, after autoclave, you can fill your reactor with media cells and just press the start button to get going. So very, very quick to get real results out of the Maverick.
Thank you so much, Chris. I really appreciate the explanation of what is in the box of Maverick. So at the end of the day, I think everyone would be interested in understand, what the data looks like. So maybe you can share with us a little bit of data around how the data generated by Maverick aligns with more traditional offline analyzers. And also maybe, I think that would be great.
Sure. Yeah, so we brought in some data, at least for the purposes of the webinar, and we'll certainly have more to share in some application notes. But so one group of studies were spike recovery studies. So we've designed Maverick to work across a really wide variety of media formulations and bioprocesses. So spike recovery is, of course, a gold standard in analytical sensitivity and selectivity evaluation. So we did some CHO optimized medias, HEC, as well as some stem and T cell and other media formulations. And so these are spiking studies where independent analytes are spiked in, and you can look and follow that the response of the de novo model on the Maverick seems to follow what would be expected based on the spike. And it also allows you to calculate selectivity in a way that's consistent with regulatory guidelines and IUPAC standards. So this is a short view of those spiking studies, at least rolled up in summary. So we did fresh media spiking. Those are the two plots on the top. We also did some spent media spiking, and those are on the bottom. So when we're dealing with fresh media, that media sometimes comes with glucose in it. So the reference glucose here was assessed using an offline analyzer, and then the spike amounts are pre-described by the experiment. So a specific amount of glucose was added to these media, and then we saw what Maverick was actually reporting versus what the reference analyzer was seeing. So CHO, HEC, stem cell, another, as noted here, and you see we have a nice linear response to those spikes. It's very, very close to a slope of 1, which is what you'd love to see, and a good correlation across those spikes. In terms of lactate, same is true. So fresh media doesn't contain lactate by design, and so the spike values are just on the x-axis there. But same thing goes, slope very, very close to 1 with a nice correlation coefficient. Spent media analysis, of course, may have some amount of glucose, some amount of lactate. So once again, a reference analyzer was used to quantify the amount of glucose and lactate that was present and compared to Maverick's reported glucose and lactate. And here again, you see a nice response with a slope that's very, very close to 1, which is excellent, and very good correlation coefficients. In terms of the actual recovery across different spike levels, so we did 2, 4, and 6 gram per liter spikes across 17 formulations in this study, and we did 1.5, 3, and 4.5 gram per liter spikes of lactate. And regardless of the spike level, we saw recoveries that were very close to 100% across the board, which is very good.
Indeed, yes. Now, let me ask you what I think a lot of bioprocess scientists and engineers may have on their mind. Have you run Maverick in actual bioprocesses, maybe CHO or other cell line, and how does the data look like?
Yeah, we've certainly run a lot of bioprocesses with Maverick. And as noted, the focus was on generalizability. So in validation of this de novo model and the Maverick performance, we've looked at a lot of media types. a long list there and still going. Also looked across cell lines and multiple varieties of those cell lines, and multiple bioreactor scales. So this is sort of the quick summary of bioreactor scales, cell lines, and media types. We'll be continuing to update these. This is an overview of the validation runs across multiple locations. So certainly we've run at 908 Devices. We have a bioprocessing lab with multiple reactors, reactor types. In validation, we've done actually more than 12 runs at this stage. Culture Bio, which is a group I'll talk about shortly. We've done 2 runs over there. CPI in the UK, 4 runs, some at UMass Lowell with Professor Sun-Kyung Yoon, and more at 908 Devices that were HEC 293 as well. So a wide variety of conditions, reactor sizes, cell lines. We chose 2 to highlight here. So one was with the group at Culture, and Culture is a company in the Bay Area, and they have some very interesting capability that they've been developing over the last several years. So they provide an integrated platform of bioreactors and cloud-based software, simulation tools, and services to try to make process development faster and more collaborative and predictive for their customers. And they developed a cloud-based application that can govern and run its bioreactor fleet. That software is called Console, and it's a really neat capability. And we're really pleased to work with them for sort of real-time monitoring and observation of the bioreactors. So they had 2 Mavericks there. And in terms of summary for these particular runs I'm going to share, so it was a NIST-CHO run. There's a lot of sort of details that I won't get into on this slide, but they have a 250 ml baffled mammalian reactors. We ran 4 bioprocesses here. So 2 of those were being monitored by Maverick, and 2 of those were essentially control. So we wanted to make sure that the presence of the Maverick probes didn't either disturb cell growth or titer. And we used a CHO-fed basal media and had a feeding strategy along the way that you'll see. In terms of analytics, so Maverick, 2 Mavericks, as noted, we're monitoring glucose, lactate, and biomass. They also have other analytics that are available in real time, so pH probe, dissolved oxygen. Offline, a bioprofile Flex 2, Bicell, and a CDEX for IgG titer. So all this information was collected. This is a look at this Culture Console. So that's what they call their software for viewing remotely the progress of these bioprocesses, as noted here. So they have many bioreactors at Culture. We were using bays 288 and 89 for the processes that Maverick was monitoring, and then they had 2 that were running as controls. So same process, same feeding strategy, timing, but we wanted to make sure that Maverick wasn't disrupting cell growth. And as you can see here, in terms of the controls versus Maverick, no difference effectively in total cell density between the controls and the Maverick monitored reactors, and likewise for titer, which was great. And this is the data that Maverick was acquiring. So glucose was on a regular feeding cadence. As noted here, they were monitoring glucose and lactate with an offline flex. And yeah, Maverick trucked along really, really nicely. So you can see the infrequent monitoring with the offline method, but as soon as you had a feed, Maverick was tracking the resultant step feed, just like you would expect. And lactate following along very nicely across those 2 reactors. And in terms of Maverick's biomass unit estimate, so the biomass units tracked along very, very nicely with total cell density, which is what we would expect in both those reactions. We also did a HEC, which is very interesting, at UMass Lowell, as noted, and the experimental conditions are here. The net is we were using 3-liter bioreactor vessels here, and a couple of different cell culture media types that we were running. So we did have glucose and lactate on the flex. In some cases, we also had YSI available, and total cell density and viability were collected with a cell count. So on the top here, so this is HEC 293 in freestyle F17 media. In this case, as you can see, we had both flex and YSI for glucose and lactate. And that's always very informative because the enzymatic analyzers, while they're quite precise, certainly they do have a tendency to drift around with respect to their calibration. So it's nice to have two independent reference values here. And Maverick followed along really nicely. They actually almost entirely depleted glucose at one stage and gave it a large step feed, but a very nice agreement with both flex and YSI. And likewise, so total cell densities aren't extraordinarily high in HEC 293, but this one got up to around 4 million cells per mil, and Maverick's biomass units tracked reported values quite nicely. And down below is HEC 293 with a different media type. We didn't have YSI in this case. We just had flex, but once again, nice trending of Maverick's results with the offline values as well as for the biomass units. You can, of course, convert. I noted that Maverick reports biomass units. That's essentially like an optical density or a capacitance reading. So it doesn't produce absolute total cell density values, but given the linearity between the two, you can convert them with a conversion factor. And Maverick's screen allows you to enter a conversion factor, so it will report in million cells per mil. And this is just an example of that. Usually with a single value, you can get a nice conversion efficiently given the linearity. And so this is taking a reference TCD and converting Maverick's biomass units to estimated TCD. Get very, very nice tracking here with a show run.
Thank you, Chris. It was really good to see how the Maverick worked in real life, in real bioprocessing runs, and how well it tracked with more typical offline analyzers. So thank you for that. Looks like you and your team did a lot of work there. So in summary, basically the Maverick shown here again as the inline analyzer of the 21st century, basically can be used to monitor multiple bioprocess parameters right out of the box. No modeling required. But if someone wants to do more modeling for more parameters, definitely the Raman spectral data is available. Also, the Maverick can be connected with up to 6 bioreactors from a single hub, which is great. And then communication via analog or OPC UA enables feedback control loops and integration with data acquisition systems. The Maverick is 21 CFR Part 11 compliance ready, which facilitates tech transfer into potential GMP environment. And finally, I know that you and your team will continue to develop more models to evolve and add other process parameters and tools in future software upgrades. We will be at the Bioprocess International Conference, booth 860. Please come see us. We will also have a couple of presentations, more data and more information about the Maverick. The BPI conference is in Boston, September 18th to the 21st. Come join us. We will also present the launch of the Maverick at the World Bioprocessing Summit in Berlin, September 27 to 29. And finally, you can learn more about the Maverick on our website, listed on this slide here. So thank you so much, Chris. And thank you for anyone joining. We can now answer any questions.
Yeah, awesome. Graziella and Chris, thanks so much. This was truly some innovative stuff. Fascinating to see, a lot of great stuff coming out. And I'm sure our audience is definitely interested in a lot of stuff, and that's reflected by the questions that we have coming in. We've got time for just a couple here, so I'm going to run through those real quick. The first question I have is, could you kind of explain how the Maverick compares to other Raman spectrometers available on the market right now?
Yeah, it's an excellent spectrometer. So very, very, very comparable to commercially available platforms that are out there today. It's a great spectrometer, great resolution, deep cooled detector. So very, very low noise. So it should be entirely comparable with other platforms that are out there today.
Awesome. Thanks, Chris. So let's see. Someone else is asking, what are the first steps that they would take to set up the Maverick for inline monitoring?
Yeah, it comes down to that 1, 2, 3 slide I had. So very, very simple in terms of using those PQ standards. So there are just 2. They come in a kit. They're ready to go. There's a mode on the instrument which walks you through that PQ step by step. So there's a stand that's comes with the Maverick, as is shown in the photo under number 1. It walks you through that process. It spends about 4 minutes actually doing that check and collecting its data, and then you move it to the second tube, and it does the same, and then it issues essentially a report of the validity of the calibration, and off you go to autoclave at that stage. So, and you autoclave your probe in your bioreactor if you like, or you can do it separately, and that's all you need. Once the autoclave's done, then you're ready to roll as soon as you can get your process up and running. So it's just a start button after that.
Yeah, good stuff, Chris. I'm not sure you could have made it any easier with that. So that's perfect. All right. So you guys mentioned this, I think, once or twice in the presentation, but maybe I had a few people ask it as well. Is the Raman data, spectral data, available if I wanted to do additional modeling? Is there an easy way to access it?
Yeah. Yes, absolutely. So we recognize that a lot of groups have some process-specific elements they may be very interested in or are trying to do more, but all that's available with the raw spectral data. So at the conclusion of the bioprocess run today, the raw spectral data is exported in a double precision data file that you can take and bring it into any other package to do modeling offline. And the spectral data can come out by OPC UA as well. So you can get real-time connectivity if you're using those Mavericks as OPC UA devices. So both real-time or in one big chunk at the conclusion of the bioprocess run.
Gotcha. That's perfect. All right. And then right at the end, someone did ask one last thing here. I've got a few more questions, but I think this will be the last one we get to. We're running up on our 45-minute time limit. We might be a little over already. So they're asking, are there plans to expand to other quality attributes?
Yeah, absolutely. There are plans. So we wanted to start with something that provided a lot of really great immediate capability, and then go out and listen to the market and see what people think. But we're already investigating what should be next to add to the turnkey panel for Maverick. And so we're excited to continue to expand the platform, expand the capability with software updates, and likewise. So yeah, we should definitely be on the lookout for more capability coming down the pike.
Chris, awesome stuff. Graziella, thanks for joining us as well. This was fascinating to see, and like I said, really an innovative product. And so audience members, if you did have more questions right now, submit them through that Q&A. I'm going to pass these questions along to the team for potential follow-up. So make sure to ask them. If you don't get your question in on the right side of your screen, you should also have the contact information for all of our speakers today, in addition to myself. Feel free to reach out to us via email, connect with LinkedIn. We'll be able to get you whatever answers you need and get you connected to the right people to make sure everything gets answered. And so with that, again, thank you all for joining us today, audience members. We appreciate your time, appreciate all your great questions. And again, Chris and Graziella, thank you for joining us as well. We hope everyone has a great rest of your day, and we'll see you all next time. Bye, everybody.